This is our presentation on the effect of temperature on Vanessa cardui development by Evelyn Gonzalez, Ethan Maldonado, Axel Almanza, and Berenice Sainz.
Arthropods are a very diverse group in the animal kingdom that include animals such as insects, crustaceans, spiders, scorpions, and butterflies. The painted lady butterfly is one of many that migrates very long distances. Some travel as far as from North Africa or the Middle East all the way to the mainland Europe to even Britain and Ireland. This species of butterfly likes to habitat places that are open and dry, but they can also be seen pretty much anywhere in the year. There are different factors that can affect the growth of the larvae, which is why it is important for the butterflies to migrate to a place that best suits their needs and can provide them with the proper setting for them to prosper. The ideal temperature for a larvae to successfully complete metamorphosis is between 26 and 30 degrees Celsius. The main purpose of this study was to observe how temperature affects the growth of the V. cardui larvae, since it has been stated that larvae growth is successful under warm temperatures. We can predict that the larvae in the group 27 degrees Celsius incubator will result in a faster growth compared to the larvae in room temperature.
So, in order to test if temperature would really affect the growth of our larvae, what we did on day one is we took measurements of both their length and weight. Once we did that, we took 2 containers and we applied 2 larvae on each container. And we decided that our standard protocol was they would get the same amount of food and the same amount of light time all throughout the 8 days, and it wouldn't change just because we wanted to reduce the chance of there being a change due to that. That if any change did occur, it was just because the temperature changed. So for food, we put about 5 to 7 grams of food, and we didn't let it go above or below that. And if it did go below, we would just add more as the week went by. And for light time, we decided to do 12 hours light, 12 hours dark time. Um, so once day one ended, we took measurements every day. And what we did all throughout the 8 days is we washed our hands and put gloves just so that no oils from our hands would transfer into the area and containers. Once we did that, what we did is we took spoons and we would pick up one larva at a time. We measured and took the weight. When measuring it, it was a bit tricky because they tend to move, so we would just make sure that they were as straight as possible when doing that. And we just repeated the same steps all across the 8 days. But on the 8th day, one of our larvae died, so we just took the measurements as if it were alive, and yeah, that's what we did.
For our results, according to the recordings, the larvae that were put under colder conditions experienced a shorter growth spurt and a shorter recorded length compared to the larvae that were put in the incubator, which was a warmer temperature. The tables to the right shows all of our recordings during the 8 days, and from these tables we can clearly see a difference in lengths and weights. For example, the larva in the incubator recorded an average length of 2.5 centimeters on the 5th day. Meanwhile, the control larva only measured an average about 0.85 centimeters. This supports our hypothesis, which stated that the larva in the incubator will show a faster growth compared to the control, which is under colder conditions. These are some pictures that we took during the experiment. On the left, we can see pictures of day 1, 3, and 8 of our control that was in the room temperature. And on the right, we can see pictures of the days 1, 2, and 3, and 8 of our experimental, which was in the incubator. The difference in size between both groups is very noticeable on day 3, where the control measured an average of 0.6 centimeters, while the experimental measured 1.2 centimeters. And day 3 is on the left, and day 3 for the experimental is on the right. So you can see the right one is like a bit bigger than the one on the left, clearly. We can also notice a difference on the metamorphic stage. Like on day 8 on the right, we can see that the experimental group started to cling on top of the container, getting ready for its pupal stage, while on the same day to the left, the control larva was still hanging out by the food and not getting ready for this next stage.
Our experiment was able to replicate findings from previous studies. In specific, research by Canelli and her colleagues found that larvae that was incubated in a warmer temperature was able to grow larger and metamorphosize faster than larvae that grew at colder temperatures. In our study, we found the same phenomenon with our control larvae at room temperature, growing at an observably slower pace than the experimental larvae, which grew at 27 degrees Celsius. So these findings can help support the observances of growth patterns at different regions and conditions. The Vanessa cardui will not grow in Canada at the same pace that it'll grow in Mexico, and this can help us explore other topics such as which species can fulfill certain ecological niches in different areas, and also what can limit or even promote the growth of Vanessa cardui in the natural world. So some limitations of our study include some common limitations that come with conducting an experiment in a classroom setting. So we didn't conduct multiple trials, and therefore we could not verify our results with precision. And in specific to our research, one of our control larvae did not make it through the full 8 days. And what this ultimately meant for us was that the data that we collected was not as rich as it could have been had we had the full sample for the full 8 days. So that's something that we can improve on in future studies. So we didn't know if the larvae died because of the temperature condition, or we didn't know if they died because of something else that we could have controlled for. So for future research, we would be interested in including more temperatures to observe the growth of Vanessa cardui. So for example, we could include a future experiment with more conditions like larvae at 30 degrees Celsius and larvae at 15 degrees Celsius. So I know I read in Canelli and her colleagues' work that they included an experimental group at 30 degrees Celsius, which didn't make it to the end of the data collection. They hypothesized that it was too warm and there might be a cap that the Vanessa cardui cannot get through. And another finding that we would like to replicate is the measurement of wingspan across different temperature conditions. The same paper by Canelli et al. Found that smaller larvae actually grew into larger butterflies, and they had larger wingspans as compared to the Vanessa cardui specimen that were larger in the larval stage. And this finding was so interesting to me, and I think we would really like to follow the larvae into the adult stage of life so we can see if these observances are replicable.
Revisiting our purpose for this lab, we wanted to know if temperature affects the growth of the Vanessa cardui larva to further educate ourselves and others. Our research could be helpful to many people who may be interested in raising moths or butterflies who are wondering what temperature could result in a successful full growth. In this lab, we experimented on a total of 4 larvae and separated them into 2 groups, our control, it being room temperature, and our experimental, it being 27 degrees Celsius, which was warmer than the room temperature. We then measured and weighed the larvae for a total of 8 days. Unfortunately, we did encounter a death of a larvae in the control group, but this error only brings more solutions to our mistakes. In the end, we can say that our hypothesis was supported by our results. Since our goal was to find out whether larvae would grow faster in a cold or warm environment, we did a bit of preliminary research, and this resulted with a hypothesis that stated that larvae would grow faster in warmer environments and will show a faster growth rate compared to colder environments. This can be supported with our results that show difference in lengths and weight between the groups as shown in previous slides and data tables. We also looked into the past research by Canelli that found that temperature had an observable effect that translates into adult phase of Vanessa cardui, with the specimen that were kept at lower temperatures producing butterflies that were larger in wingspan, and with this information we can then support our hypothesis even further. In the future, it's possible to take this knowledge and research how the temperature can affect other factors later on in the larvae metamorphic cycle that our group did not experiment, such as the pupa stage, and absorb the wingspan measurements. And these are just the works cited that we use for all of our research. Thank you for listening to our presentation. I hope all of this helped some of you guys. Our results were helpful for you. Thank you and have a nice day.